let's talk about how HeadaTerm 2 works to provide migraine relief without medication. So we start by placing the device on the forehead. Let's say this is a face here. I'll get into more detail on how to turn it on and its functionality in other videos, but this is going to start our journey. On the forehead and everywhere in our skin, there exist mechanoreceptors. So let me just draw a face here. Nose, chin. Okay. So there exist mechanoreceptors. And these turn mechanical energy, like touch, into electrical energy. So an action potential, okay? Very similar to what's on the logo for the HeadaTerm 2 device. We'll get into action potentials later, specifically when we talk about cortical spreading depression, but not for HeadaTerm. Furthermore, there are also nociceptors that sense pain. So nociceptors are free nerve endings, represented by this. They respond to noxious stimuli. And the stimulus can be anything that may cause injury. So a paper cut or a burn, for example. So these neurons are the most basic unit of the nervous system. Specifically, they're pseudo-unipolar neurons. So cell body, one axon splits into two branches. And we see these neurons in most places where the body collects sensory information, including the cranial nerve we care about for this lecture, which is cranial nerve 5, also known as the trigeminal nerve. Very important. Cranial nerve 5 is the trigeminal nerve. So as a device activates, it generates a series of electrical pulses. And these pulses will have a specific frequency. It will have a specific pulse width. And it will have a specific intensity. This specific combination of frequency, pulse width, and intensity is crucial to the way it will relieve migraine pain. So these electrical pulses from the HeadaTerm 2 device will first encounter the skin, right? So it'll first encounter the epidermis, it'll pass through the dermis, and their target is just beneath, which are the supraorbital and the supratrochlear nerves. So these nerves are actually branches of the ophthalmic division, V1, of the trigeminal ganglion. So let's draw the trigeminal ganglion here. Comes in. Again, they're pseudo-unipolar neurons, so the cell body is here. So they're in the trigeminal ganglia. So let's just label it TG, okay? Now the trigeminal ganglion has three main divisions. So it has V2 and V3. So V2 and V3. So V1 is the ophthalmic nerve, V2 is the maxillary nerve, and V3 is the mandibular nerve. The ophthalmic nerve, the one we want to focus on, V1, innervates the forehead, the dorsum of the nose, upper eyelid, orbit, including the cornea and conjunctiva, and other things that aren't visible in this drawing, like the mucous membranes of the nasal vestibule and frontal sinus and cranial dura. And then V2 and V3. So when the HeadaTerm 2, when the device activates, it's on the forehead. So what we care about is the V1 division of the trigeminal ganglion. And they represent a gateway to the trigeminal vascular system, which is an important part of migraine pathophysiology. So I very much recommend this book. And migraine pathophysiology, trigeminal vascular system activation. Very important part of migraine pathophysiology. So the nociceptors that are activated can be A delta fibers, or they can be C fibers. But we don't want to cause pain, right? That's what nociceptors are. They sense pain and temperature. We don't want to cause pain. We don't want to activate HeadaTerm 2 and cause pain. So what's lovely about HeadaTerm 2 is that it activates another fiber called A beta fibers. So A beta fibers. So we have A delta fibers, we have C fibers, and we have A beta fibers. A delta and C fibers are your nociceptor fibers. A beta fibers are your touch. So A delta fibers, they generally have a small diameter and a very thin myelin sheath. So they're thinly myelinated. C fibers have an even smaller diameter and no myelin whatsoever. A beta fibers have a very large diameter and they are very highly myelinated. So I said that A delta and C fibers respond to pain and temperature, and that's true. However, A delta is considered to be fast and C fibers are considered to be slow. What does that mean? So let's say if you hit your forehead, A delta fibers will be quick to respond. It'll give you a sharp, highly localized, painful sensation. But afterwards, you'll feel this dull throbbing and poorly localized sensation, and that would be your C fibers. So A delta, fast, C fibers, slow. And we recognize this. If we hit any body part, A delta fibers react quickly, highly localized, sharp pain, and then afterwards we get this dull, poorly localized, throbbing pain, and those are your C fibers. And this lasts longer than your A delta fibers. As I said, A beta fibers don't respond to pain, but they respond to touch and pressure. And this is what HeadaTerm 2 mostly activates. It sends an electrical signal that activates these A beta fibers. So it doesn't feel like pain, but touch. What's great about HeadaTerm 2 is that you actually have 9 levels, so you can control the intensity. So there's 9 levels here. And that's really important because if A delta nociceptors start being recruited, meaning you feel pain, you can just go down a level and have the maximum impact with no pain. And it works because A beta fibers have the lowest activation threshold. What that means is, let's say there's an action potential here. Let's say this is the membrane potential, MP, in millivolts, and the x-axis is time. Okay, so the y-axis is millivolts, the x-axis is time, and neurons are generally at negative 70 millivolts, and threshold to be activated, let's say it's negative 50, right? So for a neuron to be activated, it must depolarize. In other words, it has to become more positive, but it must reach a minimum threshold for this action potential, this signal, to go propagate through the neuron. So A beta fibers have less of a threshold. So instead of negative 50, let's say the threshold would be negative 60 millivolts. So A beta fibers, so you have a resting membrane potential. It reaches the negative 60 threshold, which means an action potential can propagate. So negative 60 would be the threshold. Again, this is just an example. Whereas for an A delta fiber, it would need to reach negative 60 for the action potential. So A beta fibers, again, have less of a threshold, meaning the threshold is more negative, so it requires less of an influx of positive ions to activate the neuron, so it can be activated and recruited without feeling pain. So A beta fibers are recruited at a lower intensity. If A delta fibers were recruited at a lower intensity, you'd feel pain first before you feel touch. So obviously that doesn't make sense practically. If you put a thumbtack on your forehead, for example, and you lightly touch it, it'll feel like slight touch. So mostly A beta fibers are being activated. They're being recruited because they require less of a threshold to activate. But if you push harder, you reach the threshold where nociceptors are activated, like A delta and C fibers. And this is why we can be specific in regards to which fibers we recruit with HeadaTerm 2. Also, A beta fibers respond preferentially to high frequency. They respond preferentially to high frequency. And this is what HeadaTerm 2 delivers, with a more rapid and repeated stimulation. The pulse width is also long enough to effectively depolarize or activate A beta fibers, but short enough to avoid sustained activation of nociceptor fibers. It's really awesome how we can use science to determine the correct parameters to activate different fibers. And this selective activation is crucial because it sets the stage for the pain modulating effects to come. So let me erase all this. So these electrical signals from HeadaTerm 2, now transformed into neural impulses, enter the trigeminal ganglia. This would be V1. Again, trigeminal ganglia is just a cluster of cell bodies that sits right outside the brainstem. So from here, they go into the brainstem. Let me draw the brainstem here, the midbrain, the pons, and the medulla. Okay, so it enters something called the principal sensory nucleus of cranial nerve 5. So let's draw that here. So this is the principal sensory nucleus of cranial nerve 5. Meanwhile, A delta and C fibers to send down the spinal trigeminal tract to the spinal trigeminal nucleus. Actually, let me change the color here. To the spinal trigeminal nucleus. So this would be the spinal trigeminal nucleus. Spinal trigeminal nucleus. So this is the medulla. It actually goes down to C3. So pain and temperature fibers descend while touch or sensory fibers go to the adjacent nucleus. There are also other nuclei. There's the mesencephalic nucleus and the motor nucleus, but that's not part of our discussion. What we care about is the spinal trigeminal nucleus. Now, while the A beta fibers, or many of them, these low threshold mechanoreceptors, go to the sensory nucleus, the principal sensory nucleus of cranial nerve 5, some of them will also transverse down and go to the spinal trigeminal nucleus as well. Now, when these fibers arrive at the spinal trigeminal nucleus, they synapse with second order fibers. So these are first order fibers. So first order fibers, and they synapse with second order fibers. The entire pathway is only 3 neurons until you process pain. The second order neuron decussates, also known as crosses over the midline, and it goes up the ventral trigeminal thalamic tract and ends up in the thalamus. So let's draw the first order neuron here, and then a second order neuron here. It goes up the ventral trigeminal thalamic tract if it's in the STN, spinal trigeminal nucleus, and it ends up in the thalamus. Let me just label this as STN. Okay, spinal trigeminal nucleus, and it makes its way. This is the brain cortex, let's say. This is the thalamus. Okay, and it makes its way to the VPM of the thalamus. And then, so the thalamus is like a relay station or sorting station. It sends pain signals to the appropriate part of the brain, the cortex that correlates with that specific body part and sensation. So the second order neuron synapses with the third order neuron in the thalamus, and it goes to the appropriate part of the cortex. This is where we finally consciously process pain. This is our brain, by the way. I'm sure you can't tell by the drawing, but that's the brain. And this is the cortex. And this is where we finally consciously process pain. Now, let's say this is an A delta fiber right here. So this is an A delta fiber. And I said some A beta fibers also go to the STN. So A beta fibers. So these A beta fibers actually trigger inhibitory interneurons. They trigger these inhibitory interneurons, and these interneurons, using neurotransmitters like GABA, so this is a second order neuron, and this is a first order neuron and first order neuron. So these interneurons dampen the activity of the pain transmitting secondary neurons. And how do they do this? Via an interneuron. So an A beta fiber sends a signal to an interneuron that synapses with a second order pain neuron. It releases neurotransmitters like GABA to dampen the signal of the second order neuron. And this is the first line of defense for HeadaTerm 2. This occurs in milliseconds of the initial stimulation of HeadaTerm 2. But that's not all. As our secondary neuron ascends, it lets off collaterals. So it sends signals to various structures along the way. I won't draw it, I'll just write it so it doesn't get too messy. So there are 3 structures we care about. Sends to the reticular formation. And this is important for arousal, and that's why it may be hard to sleep during a migraine attack. It sends information to the hypothalamus. And this results in a stress response, and it sends information to the amygdala. So with the amygdala, it may result in anxiety or depression during a migraine attack. So as pain ascends, it also lets off collaterals. C fibers would let off collaterals to the reticular formation, the hypothalamus, and the amygdala. Now, the signal from HeadaTerm 2 modulates these collaterals. Now, we don't know exactly how, but HeadaTerm 2 also modulates the periaqueductal gray. Now, the aqueductal gray is located in the midbrain, so let's just put it here. Okay. This is not to scale, obviously. Let me just put it here and write periaqueductal gray. So it's located in the midbrain, and it's around the cerebral aqueduct. So the periaqueductal gray is like a control center. It receives inputs not just from the ascending pain signals, but it also receives inputs from higher brain areas. So the periaqueductal gray will receive inputs from the cortex, receive inputs from the amygdala, receives inputs from the hypothalamus, receives inputs from the reticular formation. Now, this convergence of information allows the PAG to integrate sensory, cognitive, and emotional aspects of pain. So when it's activated by these inputs, it sends the signal down to the rostral ventromedial medulla, or the RVM. The RVM, sitting in the brainstem, contains several important groups of neurons, including the Raphe nuclei. When the RVM receives signals from the PAG, the Raphe nuclei sends serotonergic neurons. So it releases serotonin, and it synapses with another neuron that releases something called enkephalin. Enkephalin is an endogenous opioid. So the enkephalin synapses with the second order pain neuron, and it stops it from sending a pain signal. Now the PAG will also send to the locus coeruleus. This is also in the brainstem, and it will release norepinephrine. Let me just write it as norepi here. So all of this together creates a powerful inhibitory effect. The PAG and the RVM initiate a top-down pain control effect. They send signals cascading back down, creating a descending pain inhibition system that counteracts migraine pain. So while HeadaTerm 2 targets pain, it also impacts multiple migraine symptoms that may accompany the pain. So the surge of norepi enhances alertness and focus, clears that cognitive fog that may accompany migraine attacks. The serotonin synthesis, there may be an improvement of mood because of this, a decrease in anxiety. It may also provide a crucial role in regulating sleep patterns, potentially addressing insomnia during a migraine attack. There is also an increase of endorphins. Another endogenous opioid just attaches to a different receptor than the enkephalin. Endorphins attach to the mu receptor, and enkephalins attach to the delta receptor. So endorphins, they not only alleviate pain, but they also raise the migraine threshold. Okay, remember the action potential chart? Instead of here, it would need to be activated here. So you need a lot more positive ions going into the neuron for activation. Okay? So what does that mean? It makes the brain less susceptible to recurrence. GABA activity also increases. There's an increase of GABA, and there's a decrease of the neuroexcitatory neurotransmitter glutamate. This rebalancing helps the neuronal hyperexcitability associated with migraine attacks, because we do not want the threshold to be low. We want to increase that threshold, so it takes a lot more stimulus for the action potential. We won't feel pain without the action potential. We need the action potential. We need the electrical signal to go through these three neurons so we can consciously, unfortunately, process pain in the cortex. Without this signal, we won't feel pain. And therefore, if we increase the threshold, we prevent this electrical signal from traveling up this pathway. So we have modulation at the cortex because of HeadaTerm 2. We have modulation at the second order neuron. We have modulation at the reticular formation. We have modulation at the hypothalamus. We have modulation at the amygdala. We have modulation at the periaqueductal gray. We have modulation on the RVM. There is modulation everywhere because of HeadaTerm 2. Trigeminal nerve itself, cranial nerve 5, has modulation because of HeadaTerm 2. It results in the reduction of inflammatory neuropeptides like CGRP, PACAP, so it leads to reduced neurogenic inflammation. Less inflammation, meaning threshold increases. There's less of a stimulus. So essentially, this entire process is becoming less active. And the effects aren't even transient. The more HeadaTerm 2 is used, the more neurochemical changes, altered pain processing pathways, and normalized brain activity patterns that persist beyond the treatment session. So that's why it can be used for preventive and abortive. All of this is kind of like reprogramming the nervous system, reprogramming the brain and the pain processing pathways to make it less susceptible to migraine attack. Because our nervous system has neuroplasticity, it could also explain the long-term effects observed in many patients that use HeadaTerm 2, that use this ETNS, this external trigeminal nerve stimulator. It's not just treating the symptoms, it's potentially addressing the underlying susceptibility to migraine attacks. And this is how HeadaTerm 2 works.